Hello, everybody. Welcome to the second webinar of the PHARMECO IHI project about innovations in biomanufacturing and decontamination. I welcome you today for three different presentations about our project. And we will today share a little bit of insights, what we're planning to work on in this project. And I will, in a second, present the three speakers to you. So first of all, I would give you a brief introduction into the PHARMECO project. Next slide, please. PHARMECO is a publicly funded European project in the framework of the Innovative Health Initiative, IHI. It's a public-private partnership advancing safe and sustainable by design practices in pharmaceutical manufacturing, of small molecules, biologic APIs, and tides, so nucleotides and other tides, as well as all aspects around decontamination, sterilization, cleaning. So it's planned to spend most of the efforts on implementation of new technology, to implement technological innovations, scale up in pharmaceutical manufacturing, as well as quantification of these assessment of these new innovations by new standardized sustainability assessment methods. The consortium comprises 31 international partners, out of which 18 are public partners, small and medium companies, and 13 are big pharma or medtech companies as private members of this consortium. The consortium is coordinated, that means led generally by the University of Ghent, represented by Sarah Costas, the coordinator of this project, as well as the project leader, represented by myself at Sanofi in Germany. The total budget of the project is 45 million, of which half is funded in cash by the European Union and the other half by the private members of IHI joint undertaking. The project will be for 6 years, ending end of October 2030. Next slide, please. Today we will present you the work that we're planning and some ideas on existing pre-work already about three topics. We'll start with chemical synthesis in water as a reaction medium, which is one major topic of research in this project. Our second speaker will talk about innovation in biomanufacturing, which covers more the biomanufacturing part of biological drugs and biomolecules. And the third speaker will present you new technology about decontamination using supercritical CO2. At the end, we'll have some time for questions and answers. Please use the chat to make any comments or to raise your questions. And with that, I hand over to Wilfried Brahe from AbbVie in Germany and to Sándor Eötvös of RCPE in Austria, who are the work package leaders of work package 3 and present to you news about the chemical synthesis in water as a reaction medium.
Thanks, Christian, for the kind introduction. Can you go please to the next slide? Before we get started with the chemistry in water case study, which is presented by Sándor Eötvös, I would like to show you a little bit of the work package 3, the chemical synthesis platform, what we are doing there. So basically our mission is to develop sustainable and scalable methods for synthesizing APIs and intermediates. And also we like to focus thereby using green chemistry technologies with the ultimate goal to reduce waste, hazardous substances, and minimize the environmental impact. As you can see, it's a rather complex and full-blown project already. We have 6 sub-projects. The first one is on biorenewables and non-precious metal catalysis. The second topic is on key reactions in water. And as mentioned before, Sándor will share a really great case study on this topic. The sub-project 3 is the extrusion-based solvent-free protocols, which basically entails mechanochemistry. Then as the 4th sub-project, we have photo and electrochemistry. As the 5th sub-project, it's biocatalysis. And as the 6th sub-project, we have peptides and oligonucleotide synthesis. And as you can see on the right, we have a nice mix between academic partners from various institutes and universities, always having the combination with a co-lead from industry. The overall project leaders are Professor Oliver Kappe from the University of Graz and Sándor Eötvös, who will now present you the case study.
Thanks a lot, Wilfried, and thank you very much, Christian, also for the kind introduction. So on the next slide, I just want to share some brief introduction and why we want to deal with water as a substitute for organic solvents. So basically, one of the key goals of PHARMECO to reduce the amount of organic solvents used. And I mean, this is quite easy to see why this goal is important. As you can see in this slide, there is a lot of benefits, of course, of using solvents, but unfortunately, there are very strong disadvantages also. It's not only, of course, toxicity of some of the solvents, such as DMF, NMP, DCM, etc. But also it is very important that using solvents is a huge cost for the manufacturing. And then, of course, a quite large amount of organic waste generated during synthesis, especially in the pharma industry, is basically solvent waste. So it is quite clear that we do need something instead of organic solvents, and given the fact that water is available, it is cheap, it's non-toxic. It's quite straightforward to think about water as an alternative to organic solvents. Unfortunately, the picture is not that simple because there are multiple disadvantages, mainly technical limitations, and in my opinion, the most important is mainly the limited solubility of typical organic compounds in water as reaction medium. If we go to the next slide, we can see that basically the application of surfactants is a quite widespread strategy to overcome such a limitation. If we employ surfactants as, I would say, additives for chemical reactions in water, basically what is happening that we form micelles. And basically, we can take these micelles as nanoreactors, I would say, and in such instances, the actual chemical reactions, even the water-sensitive ones, take place inside the micelles. So actually, this field was sort of flourishing in the past 10, 15 years, and a lot of chemistries have been explored and exemplified under such conditions, and accordingly, novel surfactants have been developed, and some of these surfactants were designed for specific chemistries in mind. Unfortunately, such reaction mixtures usually tend to be multiphasic, so therefore slurry formation and limited mass transfer and difficult to mix reaction mixtures is the most important technical limitation in this experimentation strategy. If we go to the next slide, I just want to briefly show you another strategy. Basically, some of the surfactants, typically the designer surfactants, what are sort of developed for specific reaction types, it might tend to be quite expensive. And it's a significant drawback when we are speaking about potential manufacturing solutions. So therefore, there is a constant need for effective, but still cheaper, more cost-effective alternatives to those designer surfactants. And actually, we got very excited about some literature findings where researchers found very simple polymers, for example, hydroxypropyl methylcellulose, in short HPMC, quite effective in enabling water chemistry transformations. So Basically, HPMC by definition, well, I cannot say it's not a surfactant. I would say it's a surface active additive, but the working principle is pretty much the same as surfactants and the formed micelles. So in case of HPMC, what is happening, some hydrophobic pockets are formed, as you can see in this slide. And basically, the reactions, even the water-sensitive ones, readily take place inside these pockets. Unfortunately, not only the working principle, but also the limitation is similar to the application of micellar chemistry, because of the fact that aqueous HPMC mixtures, especially in the case of the application of solid catalysts, etc., tend to form slurries. And another addition, basically, is the fact that different HPMC aqueous solutions at different temperatures tend to form gels. So handling HPMC, aqueous HPMC mixtures as reaction solution is quite challenging technically. So if we go to the next slide, I would like to a little bit highlight the limitations of chemistry in water. And in order to do so, I just want to highlight an excellent publication recently published in OPRD. Basically, the authors of this publication done an excellent job to collect and categorize recently published water chemistry transformations. And as you can see here, as a conclusion, they basically built up 4 categories, but unfortunately, many of the explored transformations were actually quite challenging to reproduce, especially reproduce at larger scales. And it's actually quite disappointing that, for example, amidations, cross-coupling, nitroreductions, which are, of course, very important and very solid transformations for the pharma industry, even though different surfactants or enablers were present, were quite difficult to scale when using water as a reaction mixture. So if we go to the next slide, in order to demonstrate a bit more practically these limitations, I just want to show you one very simple experiment. Actually, we've done this experiment, this series of experiments a couple of years ago in our labs in Graz at RCP. And what we done, we basically took a very simple reductive amination. Also, we took some literature conditions. So as you can see, we use TPGS 750M, which is a very well-known designer surfactant. We use palladium charcoal as heterogeneous catalyst, and also there is a hydrogen source for the reaction. And what we done, we simply perform the reaction at the different scales. As you can see, in a very small scale, basically, this is HPLC vials. The reaction works like a dream, so very high yields achieved. But when we try to increase the scale, still speaking about relatively small lab scale experiments, there is a significant drop in the conversion, even though we were trying to increase stirring rate. And this is a clear demonstration, in our opinion, of the limitation, particularly the mass transfer limitation of organic chemistry transformations in water. So if we go to the next slide, basically this is the starting point of our sort of proposal, how to get over this limitation, especially thinking about scalability. So our idea was to make use of instrumentation, flow chemistry instrumentation specialized for slurry handling. And yeah, of course, normally people dealing with flow chemistry want to avoid precipitation and solid forming, but gladly nowadays there are quite a broad array of manufacturers and different instrumentation particularly designed to enable slurry handling. So basically, we teamed up with CreaFlow, who were kind enough to provide us their HANU reactor. Without going into too much details, the working concept of the system is the beneficial combination of active and passive mixing. Well, passive mixing was simply achieved by a series of cubic mixing elements. You can see enlarged on the top right corner of the image, while active mixing was given by an oscillator pump, and the oscillator pump together with the cubic mixing elements allowed an excellent, robust slurry handling and also a very good mass transfer alongside the reactor. So on the next slide, I want to show you a case study. Basically, this was our first entry into the field of chemistry in water using flow chemistry instrumentation. So the model reaction selected was the reductive amination. This is the best conditions shown here on this slide. So we had to optimize apparently the usual conditions for flow chemistry, such as temperatures, residence times, pressures. But it was actually very interesting to see not only these usual conditions, but for example, oscillation parameters, amplitude and frequency of the oscillator pump had quite a significant effect on the reaction outcome. We were quite happy to find good productivities and a very low effector for this experiment. And it was also quite important that we were able to reuse and recycle not only the heterogeneous catalyst, but also the surfactant solution, which is important given the fact that TPGS 750 is not cheap. So if we go to the next slide, I just want to quickly introduce another example, little bit considering scalability more in this case. This case is the SNAR reactions, also very important transformation for the pharmaceutical industry. And we use the same CreaFlow instrument, but this time, basically, we performed the direct, I would say, mini scale up within our labs. So all the optimization was done with the smallest scale, the 5 mil instrumentation, and this basically allowed us the use of minimal amounts of reagent volumes for parameter optimization. Then conditions were directly scaled in a 15 mil reactor. Actually, the benefit of this system is that most crucial physical parameters such as channel dimensions, cubic mixing element parameters, their size, they were totally the same. It is only the length and thereby the volume of the reactor what is different. So we only had to increase the flow rate to have residence time fixed from the initial optimizations, and no further optimization was required between the scale-up. And actually, this is also possible in a pilot scale instrument, what is available from CreaFlow. So basically, the best conditions shown for two selected examples. Again, you can see good productivities, 5 grams per hour from a small footprint system, I think is a good output. So if we go to the next slide, this is basically our current focus, amide bond formations. Amide bond formations are also very strongly highlighted in the PHARMECO project. I think it's needless to underline and to introduce the importance of amide bond formations in the pharmaceutical industry. Actually, we have been collaborating with AbbVie and particularly with Wilfried for such a long time in this project. And I just want to share some earlier results with you. So in this project, basically our idea was not just to avoid water not just to avoid organic solvents as a reaction medium, but we also wanted to completely get rid of, for example, organic solvents during workup and isolation protocols. So with this in mind, initially we performed a quick batch preliminary screening, and we selected basically some conditions based on literature precedents, EDC and COMU as coupling agents, also together with some surfactants such as TPG, SPS, and also some more traditional surfactants such as SDS, and also HPMC was screened. As you can see from this table, basically quite good data was obtained, high conversions, good selectivity. Actually, conversion and selectivity was quite okay without any surfactant or HPMC being present. However, if you take a look to the photos, and particularly the photo in the top right, basically you can clearly see that without any enabler present, there is a clear phase separation in the reaction mixture, making a clear no-go in this experiment for further processing or process development. So on the basis of the physical behavior of the reaction mixtures, as well as the conversions and selectivity, we selected HPMC as enabler and COMU as an initial coupling agent. So if we go to the next slide, I just want to show you the flow setup we used first. This is a spinning disk reactor system. Without, again, going into too much details, the spinning disk system comprises, in this case, 3 spinning rotors, I would say. And due to the very high rotation speeds of these disks, there is very intense shear forces and very intense diffusion. And because of the fact that the components are being pumped continuously through a chamber comprising these high-speed disks, basically mass transfer is excellent in the system. And then also additional great point of the system, the reason why we selected the material of construction of the internal parts of the system was silicon carbide, chemically very much inert. You can also see some of the optimized conditions and some of the best results we achieved in the spinning this system. 25 grams per hour productivity, which gives half a kilogram if extrapolated to one day. This is quite a good result, in my opinion. On the next slide, we can see basically the next level of this experimentation. As was mentioned, we also wanted to eliminate water, organic solvents as a reactional medium, and also from the workup and purification protocol. So therefore, we had to change the chemistry from COMU coupling to EDC-mediated couplings. And given the fact that the reaction mixture, aqueous reaction mixture comprising EDC was a little bit difficult to process, a little bit more sticky, and we also wanted to focus more on a further scalability. We also had to change the reactor. This time, we used an agitated cell reactor from AM technology. The basic working principle of this system, as you can see, we have 10 interconnected CSTR zones. And all the CSTR reaction zones basically comprise an agitator. And due to the agitation, there is a very effective active mixing of the system. And if you go to the next slide and take a look a bit to the final conditions, we had to, of course, optimize concentrations to attain excellent productivities. It was also quite important to optimize HPMC content. And it was actually also important to take a look into the agitation frequency. But the very important take-home message is that under such conditions, not only the reaction, but also the workup and isolation is completely exempt from organic solvent. So basically a simple filtration and washing with water gave us pure product with excellent productivity. As you can see here, we achieved 70 grams per hour, which gives around 1.5 kilograms if extrapolated to one lab day. So finally, if we go to the next slide, Actually, we were thinking about looking for a more challenging example to conclude this project. And because of this reason, we simply opened up that nice OPRD paper, and we were looking for some sort of tricky examples. The transformation, the amide bond formation with this tryptophan derivative actually caught our attention because according to the authors, it was just a nightmare, especially to scale. So therefore, we set out to check how our conditions with the ACR reactor works. So if we go to the final slide, I just want to quickly introduce you the findings of this amidation. Conditions had to be modified a bit, but after some minor optimization, we achieved excellent stability even with this technically challenging amidation. As you can see from the results, quite good yield and also excellent productivity were achieved. And it is also very important that green chemistry metrics, most important ones, E factor and PMI, were also pretty low. So this is nicely highlighting that only a relatively low amount of waste was formed during the protocol, and definitely lower than compared to the corresponding batch study. So with that, thank you very much for your attention.
Thank you very much, Wilfried and Sandor, for this excellent presentation. We now move on to our next speaker, presenting PHARMECO innovation and biomanufacturing, covering the aspects of the biomanufacturing of drugs. And this will be presented by Simon Horden, project leader at CPI in the UK.
All right.
Sorry, Steve, sorry.
Absolutely not a problem. Before I hand over the stage to Simon, as the industry co-lead, my name is Steve Fernandez. I'm the CMC Technology and Innovation Lead for Sustainability by Design initiatives within Global Healthcare Operations business of Merck KGaA Darmstadt, Germany. And along with my co-lead, public partner Simon, co-lead Simon Horden, Chief Technologist at Center for Process Innovations Limited. We are immensely proud to present to you an exploration of intensified biomanufacturing, alternative evaluation of plant-based production of biomolecules, and the assessments of process control sustainability parameters to obtain a recombinant protein as a drug substance or a biologic API. So next slide. So we present to you here out of the 31 private and public partners, we present to you a dedicated biomanufacturing coordinated amalgamation of scientific innovators and industry implementers, bringing in a live expertise in biomanufacturing value chain. So we really cover from a choice of cells, prokaryotic, eukaryotic, and even plant-based, with bioreactor design to choice of chromatography, process analytical technology expertise, and process modeling for the next generation of digital twin designs. Next slide. So in our biomanufacturing work package, we have really separated our work package into working groups. And within those working groups, we are really addressing tasks really related to development of intensified production platforms for recombinant proteins with a novel PAT and model-based process control over CPP, including quality and sustainability metrics. We would assess the areas where the use of water for injection is essential, and optimization of energy and cost effectiveness where there is use of water. An investigation and assessment of the impact of renewables and recyclable single-use materials, which is often used in biomanufacturing as consumables, along with the biomolecule API development value chain. Model-based prediction of scaled up production performance from lab scale studies using baselines and benchmarks, then to transfer to pilot scale manufacturing processes. And all these come with a reduction in current benchmarks, and we have set certain objectives and scales where we would like to reduce 50% in energy use, 20% reduction in water use. So pretty bold overarching goals, and this is what we would like to achieve within this biomanufacturing work package. Next slide, please. So before we go into the details of what and how we would like to achieve, so this is really a generalized setup of a fed batch manufacturing using CHO cells with really an upstream bioreactor and a downstream purification unit operations. Of course, this is bioprocessing 101. And as we diligently proceed to provide a baseline in our biologics process intensification methodology, we're going to use something that's already been used by the ACS, Green Chemistry Institute Pharma Roundtable. They've implemented a metric called process mass intensity, and it clearly is defined as the total mass of input of water, raw materials, and consumables really required to make about 1 kilogram for biologic API. So the benchmark PMI for a USP and DSP, of course, already published about in 2019, where they have really calculated the PMI for an upstream processing to be close to 2100 kilogram per kilogram antibody produced, and for your DSP, it's around 6,000, which is pretty high. So can here in our partner consortium really bring down the biomanufacturing PMI by 20%. Yeah. So this is our goal and objective. So before Simon takes over in describing CPI's capabilities and public partner capabilities, next slide. I would like to share a slide that was shared by our private partner, Swift Pharma. And before I allude to the content on this slide, I would like to share that pharma R&D overall contributes to 5% of carbon dioxide. So Swift Pharma is exemplifying uniqueness to traditional biomanufacturing with environmental sustainability in mind. So this really table compares the various modes of biomanufacturing with environmental sustainability in mind. Yeah, we're going from a stainless steel batch manufacturing to a single-use perfusion to a single-use hybrid, which is a combination of fed batch and perfusion. And then finally, in the last column is their plant-based hyper-express biomanufacturing with low cap expenditure and operating expenditure costs. Well, highlighting key production cycle times, reduction by 2 to 3 weeks, from 2 to 3 weeks to mere 7 days, and technologies in downstream processing that includes expanded bed absorption and centrifugal particle chromatography. So these type of unit operations shed light on reduced centrifugation and membrane consumable use. So this is our exploration within the biomanufacturing work package to see how plant-based manufacturing really compares to your traditional CHO-based manufacturing. So now I hand it over to Simon, who would go over our public partner capabilities. Next slide, please.
Thanks, Steve. Good afternoon, everyone. I'm Simon Horden. I'm a Chief Technologist at CPI in the UK. And together with Steve, I have the privilege of being co-lead of the biomanufacturing work package for PHARMECO. So to meet our objectives and test innovative solutions across upstream and downstream processing of biologics, this biomanufacturing work package will leverage CPI's expertise and capabilities, in particular, our intensive CHO-based MAB manufacturing process flow. And we'll plug in innovative next-generation bioreactors from Cultzyme, integrate new paths such as Merck's ProCellEx Raman system, develop a robust PAT strategy and advanced process control systems, novel chromatography media from Biotoolomics, and we'll explore and test renewable, recyclable materials for consumables from Iron Plus. Next slide, please. So within the work package, we have 4 clear areas of focus. The first is advancing expression technologies, which will be led by Kultzyme, and will introduce advanced bioreactors to demonstrate the positive effects on sustainability by improving control over critical process parameters. The next area is process optimization through advanced process control and process analytical technology. This will be led by RCPE and University of Ghent, and they'll look at model-based advanced process control over critical sustainability parameters. The PAT strategy will be designed by the University of Ghent and will define the critical sustainability parameters and identify the appropriate technology to monitor them. A further area of focus will be purification. These activities will be led by biotoolomics. We're intending to look at non-capture technologies and measure the reduction in water and energy consumption resulting from that change. Finally, we'll be looking at the feasibility of using renewable recyclable materials to manufacture single-use consumables, so we'll circularize the life cycle of plastics and reduce waste. Next slide, please. So the new bioreactors from Kultzyme will aim to help the industry shorten development time by transforming the raw process data into actionable insights in real time, reducing the amount of experimental work and therefore increasing efficiency and the responsible use of raw materials. A cloud data platform will unify data from start to finish, and will try and create a digital twin to allow real-time optimization. And we'll have adaptive controls, integrated AI and sensors to adapt to process requirements. Next slide, please. Biotoolomics' innovative chromatography medium are based on shelled resins. These lend themselves to flow-through processes. The aim will be to create an entirely flow-through, non-capture process to significantly reduce the volume of buffers, therefore purified water and the energy required to produce them. Next slide, please. So using advanced control algorithms, we hope to reduce the amplitude of deviations from set points, thereby reducing the strength of the control inputs required. This will improve the efficiency of the process, reducing the requirement for raw materials while maintaining product quality. To do this, we need to define the sustainability KPIs, work out which relevant information can be measured in real time, and identify process analytical technology to enable that. We'll need to develop control algorithms based on this feedback information. So data collected will be linked to process models characterized by their time dynamic behavior. These models can then be used as virtual test beds for the controller. Based on the selection of KPIs, PAT, and process control parameters, suitable control algorithms will be chosen that can adjust process parameters in order to achieve maximum efficiency. The control algorithms will likely be model-based with optimization functions for weighting the control targets, and may be predictive to allow adaptive control based on real-time PAT. Next slide, please. So it's been shown that substituting single-use consumables for traditional stainless steel and reusable equipment brings a reduction in the volume of solvents and water used in the cleaning. This also reduces the energy requirements for production of purified water. The switch can, however, result in an increase in solid plastic waste, which is disposed of by incineration or landfill. This issue could be mitigated by the use of renewable or recyclable materials in the manufacture of the consumable. For instance, bioreactors, biocontainers for medium and buffers, membranes and capsules, sensors and housings. Next slide, please. So work package 4 integrates and coordinates within the overall project with the other work packages. By developing and demonstrating new biomanufacturing strategies, we will iteratively inform the route design process for large molecules and develop a systematic approach for the choice of biomanufacturing systems based on sustainability and economic criteria. And we'll work towards adapting the select criteria for manufacturing large molecules. We'll provide process data and modeling information to inform development of decontamination technology in Work Package 5 and allow development in the life cycle analysis in Work Package 7. And information from the demonstration runs will be used to scale up and transfer selected processes to pilot scale in Work Package 6. Next slide, please. And then I'll hand you back to Steve.
All right. So research-based evidence suggests, thanks, Simon, have suggested, and thanks to Simon for highlighting the various contributors to biomanufacturing resource consumption. Evidence suggests that biologics manufacturing process is very water intensive, about 94%. Yeah, and out of this 94%, 64% really comes from downstream unit operations, for example, chromatography. So through industry support, our public partners are geared to provide a scalable, sustainable, eco-friendly, and process-intensified biomanufacturing methods to really include the use of technology and innovation approaches, to show an impact from improvement opportunities versus the current processes that are being used. Also including recyclability of consumables used along the bioprocess value chain. So in conclusion, for the goal summary and outcome, as published in 2019, a quantitative assessment of an environmental impact of biologics manufacturing using PMI was carried out. And for better appreciation of the significance of reduction of the overall PMI for a projected monoclonal antibody demand of 200 kilograms per year, an 80% reduction in overall PMI would really conserve 19,000 metric tons of municipal water over a 10-year period, bringing to a total cost savings about 7.4 million. So the real objective and the drive for this biomanufacturing work packages can we do better again. So our drive is to reduce water consumption, and the more we reduce, the more cost effective our processes would be, and eventually it is with the patient in mind. So next slide, I would like to end by saying follow us at PHARMECO for future updates on biomanufacturing, and on behalf of the public and private partners who really provided content and contributions to this section of the webinar. Thank you all for your participation and your kind attention. Feel free to contact us via email if you have any questions. Thank you.
Thank you very much, Simon. Thank you very much, Steve, and apologies again for not for missing you in the introduction. You
all
made
good,
a great
Christian.
introduction.
All good.
Thank you very much. And with that, I hand over to Yves Bayon from Medtronic in France, who is the Work Package 5 leader of the PHARMECO project, and who's giving a presentation on decontamination using supercritical CO2. The floor is yours, Yves.
Thank you, Christian. So we will introduce you this work package on decontamination using supercritical CO2, but not only. Next slide, please. And this work package 5 has the objective mainly to reduce energy, water consumption, solvent use, and without compromising the safety, and also mitigating antimicrobial resistance, which is key. And water, as you know, is extremely consumed for steam sterilization, for cleaning as well, before and after the use of solvents, detergents, and so on. We have 4 main activities for the work package 5. A novel approach to value engineering, that means to optimally use resources for decontamination and clean disinfection. We have the cleaning in place and the sterilization in place, with the partners indicated in brackets, the photocatalytic wastewater remediation, and as well the supercritical CO2 cleaning and sterilization, which is the focus of today. Next slide, please. Supercritical CO2, what it is? It is a green solvent with some magic properties. It's not a liquid, it's not a gas, it is in between, with high density and very quick and fast diffusion, easy diffusion into materials. Can be run at low temperature, and supercritical CO2 has decontamination properties by itself and also cleaning properties. Next slide, please. Eco-friendly, so perfectly in line with the scope of PHARMECO project. No effluent generation, no pollutant generated, no residues of CO2 as well generated in the final product or extract. No waste production, non-toxic, non-flammable, safe. A lot of, I would say, very kind features. And less resources are required when you are using such a solvent. Easily available and fully recoverable as well. Next slide, please. Supercritical CO2 is not a new story. So it has started in the 70s for the decaffeination of coffee and tea, and as well used at a large scale. Pharmaceuticals in the 80s, notably for the extraction of essential oils and active compounds from plants. It is really efficient and environmentally friendly process. In the 90s, supercritical CO2 has been used for the synthesis and processing of advanced materials, including films, coating, nanoparticles, with very fine, very accurate control to achieve the materials characteristic which were desired. Textile industries, more recently, notably for dyeing, cleaning processes, offering a more sustainable alternative to comparable processing methods. And as well, including for environmental application to, for example, decontaminate soil, and for waste treatment, including the for plastic recycling. Next slide, please. This is captured in different ways. So the commercial application, extraction of molecular features, notably for the pharma industries, fragrance, aroma, still for the pharma, but also for the cosmetic industries, cleaning, including mechanical part, medical devices, plastics, notably to help the recycling, and production of aerogel, so products with very specific features. Next slide, please. Just to show you that supercritical CO2 can be run from the lab scale to the industrial scales, starting from milligram to gram of target compound isolation purification to kilogram. It is exactly the scalability which will be leveraged for the decontamination, sterilization of medical devices. Next slide, please. Once again, it is not new that supercritical CO2 can be leveraged as an alternative technique for sterilization. Not new, but recent. And not readily available at the commercial scale. That's exactly PHARMECO what will do to pave the road for more adoption by industries for decontamination sterilization. On the left side, you have seen that already a lot of materials have been sterilized or have been, I would say, tested in condition or sterilization condition using supercritical CO2. And Nova Sterilis is the first US company having developed this sterilization procedure at a commercial level with the first product approved by US and Australian regulatory agencies. Next slide, please. What will be implemented during PHARMECO is to start from this publication, quite recent, published in 2021, but to start from this optimal conditions which have been identified, particularly when supercritical CO2 is mixed with hydrogen peroxide. And you see optimal condition, 110 bar, 40 degrees, 20 minutes, 200 ppm of hydrogen peroxide. You see that the temperature is relatively low and the cycle is short as well, 20 minutes. This is exactly what we will try to go around to optimize with partners of World Package 5 for this specific task. Next slide, please. First, before introducing the partners and their roles, to have a look at equipment which will be used for supercritical CO2 technology. So don't look too much at the illustration, maybe too complex to be described in a couple of seconds. But what we will do with this, and starting with a lab scale reactor, is to play with the agitation inside the reactor, where the products will be placed for sterilization, and also to consider recirculation up, down, and down and up of supercritical CO2 loaded with hydrogen peroxide. And we will start first with a 2-liter supercritical CO2 reactor, but the design can be easily extended, scaled up to a 100-liter reactor or even beyond. And we will really investigate the role of agitation, the recirculation for optimal flow of supercritical CO2 loaded with hydrogen peroxide inside the packaging materials of medical devices to check if we can really clean and sterilize products effectively. And then this will help the design of the industrial scale reactor. Next slide, please. Partners, right, co-leading this task with Sarodas Petronis, and also co-leader of the World Package 5 with me, public research institution, and really committed to help us, starting from their know-how of development and analysis of antimicrobial materials, surface cleanliness, and characterization of medical devices. And you see that they have access to a number of critical technologies to serve our purpose. SFE process extraction, a French company specialized in the design and manufacturing of supercritical CO2 equipment for extraction, purification, cleaning, and also sterilization, one of their current focus. Medtronic, based in France, and the site in France designing, developing, manufacturing devices for hernia treatment, and also developing hemostatic products with core competences in textile engineering and biomaterials, and as well mastering the supercritical CO2 technologies at pilot and industrial reactors, notably for medical device cleaning. Next slide, please. So Medtronic will provide meshes, resorbable meshes, permanent meshes, will select packaging materials. Supercritical CO2 treatment will be done as well in the 4-liter reactor in presence of hydrogen peroxide. SFE process with 2 tasks. First, to design a specific 2-liter reactor with agitation and recirculation features, and also like a chromatographic injection loop for delicate and accurate injection of hydrogen peroxide in the supercritical CO2 reactor. To start as well series of tests to optimize the condition. So that means the pressure, the duration of cycles, hydrogen peroxide concentration in the reactor. MRIs will characterize products, meshes, and packaging materials before and after treatment to see how far we can sterilize effectively products and how far we can decontaminate. And they will run as well a series of tests with spores, gram negative and gram positive, to check the sterilization condition. Next slide, please. And that is the end of my talk. And sooner or later, you will know more about the specific supercritical CO2 activities. Thank you for your attention.
Thank you, Yves, for a great presentation. Thanks all speakers of this webinar today for their excellent presentations and for presenting the topics of our projects. And as announced, you will hear more in future webinars as well. I think we have time for a couple of questions, so please raise questions if there are any, or please share your comments, your views on the presentations. At the moment, I don't see any questions in the chat. So maybe some of you are still typing. Maybe I'll take the time to announce the next webinar. So next slide, please. We have a third webinar in our webinar series in June, which will be next Wednesday, same time, 2 to 3 o'clock Central European summertime, and which will be about steps towards sustainability assessment and regulatory preparedness. So we will answer the question how we can quantify the environmental impact of improved biomanufacturing, manufacturing and cleaning technologies. And we will also try to address issues and maybe solutions that are related to innovations in current existing manufacturing processes and the regulatory relevance of those changes. If you're interested in that webinar, you'll find the QR code with a link to our webpage and to the webinar on this page. So please register for the webinar if you are interested to join. I still don't see any questions or comments in the chat. Fanny, please, can you see any? Did you see any questions? Did you receive any comments?
No, I didn't receive anything.
Which means the presentations were totally clear to everyone. So thanks a lot for everyone presenting this afternoon. And whenever you have any comments or questions, please reach out to us through our webpage or directly to the presenters. And with that, I close the meeting for today and thank you all for joining this webinar. Thank you very much and have a good afternoon. Bye-bye.
Thank you. Bye.
Bye.